Hello, everybody. My name is Igor Grigoriev. I lead Fungal Genomics at Joint Genome Institute. I will be chairing the session tomorrow. It's my pleasure to welcome you all to the fungal session. We had biodiversity genomics meeting, and fungi include over 1 million species and over 1 billion years of evolution. We have about a couple thousand genomes already, and there are active efforts to sequence more in different parts of the world, and they produce important parts for food, medicine, bioenergy, biotechnology. We're all well aware of that, and today we have a lineup of 5 excellent talks and several posters after this session to cover fungi of different lifestyles and mycorrhiza, pathogens, ecologically industrially important species, different sampling strategies across fungal tree of life, centered around individual genus, the whole tree of life, even individual species with different types of sequencing strategies, including multi-omics. So let's start. Our first speaker is Francis Martin, Director of Center for Excellence of Advanced Research for Biology and Ecology of Forests at the France National Institute of Agriculture, Food and Environment. And he will tell us about genomics of mycorrhizal fungi. I would like, before we launch the video, I would like to remind everybody to type your questions beneath the video and vote on them. And we'll be using the most popular ones. Okay, let's start.
Okay, so today I'm going to talk about the evolution of mycorrhizal symbiosis. So in this talk of our 10 minutes talk, I'm going to summarize the work of about 50 people for 3 years, where we are focused on the evolution of mycorrhizal symbiosis. So let's go to the wood for a while. It's fall time, so that's the right time to walk in forest. And in forest, you may see 3 main groups of mushrooms, 3 main groups of fungi. The saprophytic species that you have on the left, able to decay wood. So you have the white rotors, the brown rotors, and also the soil saprophytic species able to decay litter and humus. So this is a saprophytic species. Then we have the pathogenic species that the foresters don't like so much because they are able to kill the trees, like this Armillaria. But we are mostly interested by the mycorrhizal symbionts. Here you see the iconic Amanita, which are able to talk to the trees and establish a mutualistic symbiosis with trees. So today we are mainly talking about these mycorrhizal symbionts. So this ectomycorrhizal symbiosis is well known for this ability to improve the plant nutrition. As you see on this slide on the left, these mycorrhizal symbiosis are able to establish a very extensive web of hyphae, the so-called wood wide web. And thanks to this web of hyphae, they are uptaking nitrogen and phosphates, and they are improving massively the plant nutrition. So we know that ectomycorrhizal fungi are mutualistic fungi. They play a key role in nutrient acquisition in most forest ecosystems. We know that ectomycorrhizal symbiosis emerged at least 60 times during the evolution of Basidiomycota and Ascomycota. We also know that these fungi play a key role in carbon cycling in forest ecosystems. But considerable variation exists in the capacity of these mycorrhizal fungi to acquire carbon from soil organic matter. So currently, most people think that these fungi, these ectomycorrhizal fungi, are potential organic matter decomposers, but they are not saprotrophic species. They are not able to use the released carbon for their nutrition. So in our project, we are aiming to relate genomic features to key ecological traits, for example, soil organic matter decomposition. And we have aimed to track the many independent lifestyle transitions from saprotrophism to mutualism in fungal lineages. So we mine the microcosm database at the Joint Genome Institute. You see on the left that currently there are almost 1,800 genomes stored in this microcosm database, and we have selected 135 fungal genomes, including about 70 saprotrophic, endophytic, and pathogenic species. But most importantly, we've been able to collect 60 genomes for mycorrhizal species, including about 30 new genomes. And in this project, we have sampled ecologically dominant fungal species for which there were previously no symbiotic genomes available. So this includes ectomycorrhizal species in the Russula group, in the Teleflora group, in the Cantharellus, in the Chanterelle groups. So we sequence these genomes, assemble, annotate these genomes, and we compare them in this large-scale study. So I'm going to provide you some highlights. I'm going to focus on the part of the genomes involved in the lignin and cellulose decomposition. The enzymes, so-called carbohydrate active enzymes, which are involved in the decomposition, decay of lignin and cellulose. So on this complex slide, you have on the left the number of gene copies per genome of enzymes such as class 2 lignin peroxidase, cellulase of the GH6 families, cellulase of the GH7 family. All these enzymes and the CBM1 motif involved in cellulose binding, all these enzymes are key players in the lignin and cellulose decomposition. They are needed for decomposition and decay of lignin and cellulose. And then you have the species that we investigated on the x-axis, and you see that the number of genes coding for these enzymes is very different between the different species. Some species like Galarina or Coprinus, but also orchid mycorrhizal species like Tulasnella or Sebacina, they have more than 160 genes coding for these enzymes. And so Microlepiota, Trametes, Sterium, well-known white rods, have a wide arsenal of these enzymes. CBM1, LPMOs, multi-copper oxidase, lignin peroxidase, cellulase involved in the lignin and cellulose degradation. But if you look at the mycorrhizal species, ectomycorrhizal species, on the right of the slide, you have Lacaria bicolor, Amanita muscaria, Spilus, Boletus species, and Pisoletus. You see that the number of genes is drastically decreasing and much lower. So the main finding of this large-scale genome comparison showed that the number of ligninase and cellulase, number of enzymes involved in lignin and cellulose degradation is much, much lower in ectomycorrhizal species, in symbiotic species compared to their white waters. So most of these species are depleted in peroxidase, lignin peroxidase or cellulases. And there I'm focusing on one of these clades of mycorrhizal species, the chanterelles. And so you see that on the left, within the Cantharellus, Cystostrema is a well-known white rotor, and you see that its arsenal of enzymes coding for the degradation of lignin and cellulose, CBM1, AA9, LPMOs, GH6, GH7 cellulases, carbohydrate esterase 1, there's a high copy number for these genes. Instead, in the two species of symbiotic fungi we investigated, the well-known edible hedgehog Hypnum rufescens and the golden chanterelle, you see that these two species, they encode a very low number of enzymes, genes coding for this enzyme, only one CBN1 motif and no GH6, no only one GH7 cellulase. And similarly, in the yellow chanterelle, very few genes coding for this enzyme. So there's a striking loss of hydrolytic enzymes in the symbiotic species. But we found that by looking at the early clades of ectomycorrhizal fungi, we found that not all ectomycorrhizal fungi are depopulating plant cell wall degrading enzyme, this enzyme involved in the lignin and cellulose degradation. For example, in Acephala macrosclerosioms, one of the leotomycetes, we found that there are many genes coding for these enzymes, more than 282 genes coding for plant cell wall degrading enzymes, including cellulases, pectinases, and other enzymes very efficient to decay cellulose or lignin. And then we were wondering how this fungus can enter, as you see there on the top left part of the slide, you see in green Acephala penetrating and forming the artignate in the ectomycorrhizal roots. And so we were wondering how this fungus is able to colonize the roots with such a large set of enzyme involved in the plant cell wall degradation. And so we did RNA profiling using RNA-seq, and then we found that in this species, the genes are encoded in the genome, but they are not expressed during the interaction with the plant. So this means that ectomycorrhizal fungi can control the expression of these plant cell wall degrading enzymes during the interaction with the host. So to summarize, transition from saprotrophy to ectomycorrhizal symbiosis involved at least main 4 evolutionary mechanisms: widespread losses of degrading enzyme acting on lignin and cellulose, co-option of genes present in saprotrophic species to fulfill new symbiotic function. I didn't show you data on that, but this is one of the main findings. Diversification of novel lineage-specific symbiosis-induced genes and proliferation of transposable elements. And we showed here that this evolutionary mechanism can be traced back to the early diverging clades of ectomycorrhizal fungi, like the chanterelles in the Cantarellales. So to finish, I would like to thank the people involved in this research project, people in my group, Shingo, Mayra, Annegret, Emmanuel, and myself did a lot of mining and genome analysis. In Laszlo Nage's group, Nico was a key player. And of course, I should thank my collaborator at the JGI, Igor Grigoriev, people working, annotating the carbohydrate active enzymes, Bernard Henrissat, David Ibet for the phylogenomics, and Ritas for providing many of these genomes. Thank you very much.
Okay, excellent talk. Great. I would like to remind everybody to type the questions in space below the video. Unfortunately, Francis wasn't able to join us today, but Annegret Kohler and Shingo Miyachi, 2 members of his team who worked on this project very actively, are here to answer the questions. Thank you for joining us. So maybe I'll start while I'm waiting for questions to come. So with 60 or so genomes of mycorrhiza that we have at hand, what is the next thing? do we need to sequence more? Do we need to go more into functional genomics? And given that you're part of lab that focus on biology ecology of forests, what are maybe practical translations of this research into forest management?
Yeah, so I think the very interesting thing is now that we also see ectomicrofungal genomes that are somehow in transition. So that still have the capacity, but are already interacting with plants. So what we have seen in the last slide. So I think that's very interesting clades. And I think we should focus more on these probably to understand really how the evolution goes on. And there it would also be interesting, yeah, to have a closer look into these clades. So
more genomes, that's the answer.
Not only more genomes, but also more functional analysis. That's clear, because if the genes are still there and they are regulated, then it's really, yeah, it's also the question, how is it done? And there are some of these fungi that not only form ectomycorrhizal, but other kind of interactions, and to understand how they manage to adapt to ericoid mycorrhiza or ectomycorrhizal fungi. That's very interesting, and there we need more functional analysis as well.
Great, thanks. And any practical translations into forest management? Do we have a sort of magic powder to sprinkle and?
Among these fungi are some that can probably help trees to better tolerate or better adapt to drought stress or this kind of climate changes. So yeah, we could think about, so we also learn. So what Francis presented was this loss of plant cell wall degrading enzyme, but of course we learn more from these genomes. So for example, this kind of force of mycorrhizal fungi.
Okay, all right, excellent. Thanks a lot. I think we need to start moving to the next talk. And our next speaker is Michelle O'Malley. She is a professor at the Department of Chemical Engineering at the University of California, Santa Barbara. And she'll bring us from the roots of the plants to the roots of the fungal tree of life, focusing on one of the early group of early divergent fungi, and combining genomics, multiomics, in order to understand biology of gut fungi.
Hi, everybody. My name is Michelle O'Malley, and I'm going to tell you about how we've looked at comparative genomics and other omics tools to reveal some of the interesting enzymes, transporters, and other interesting natural products that anaerobic fungi can make. And to motivate my work, really what has been at the heart of that is looking at how we can take non-food plant biomass and convert that into something value added. Many of you probably are familiar that this is called lignocellulose, and it requires a number of cellulases, hemicellulases, and ligninases to break down that polymer into bite-sized bits of sugars for microbes. And nature does this on purpose because it's trying to keep microbes and their enzymes out. And where we derive a lot of inspiration for our work is from another place in nature, which is the large herbivore digestive tract. So here you're seeing a ruminant herbivore, a cow. And why am I showing you a picture of a cow? Well, because inside of the rumen and the digestive tract of this cow are a team of microorganisms consisting of bacteria, archaea, protists, and fungi that actually work together to depolymerize that tough woody biomass into sugars and short chain fatty acids for that animal. And of course, this is a really complicated environment, and it's It's been a hotbed of research for a number of years. But what I would argue is that some of these low abundance members of the rumen microbiome, like the anaerobic fungi, are very seldom explored for their biotech potential. And so what do I mean by these anaerobic fungi? These are the Neocalamastigo shown here on the JGI microcosm fungal tree of life. There's 9, and some would argue a lot more characterized genera of these anaerobic fungi, and they reproduce not like yeast or any of the more biotech ready fungi that you might recognize, but they start from a single spore, they germinate rhizoids, and they grow a root-like system, which is thought to spit out a lot of biomass degrading enzymes that mature zoosporangium then bursts, and more zoospores are generated. And it's a really interesting system to study, but particularly because there's a lack of genomes and available strains to work with. Most people who work on these fungi are doing so by foraging and finding their own in the environment. They're difficult to cryopreserve, next to impossible to genomically manipulate, and very small community of researchers up till now has studied these fungi, but that number is growing, which is very good. And this is what they look like growing into lignocellulose. You see that invasive root-like structure. And so what we, and in fact others have done, is to go out into the world and isolate our own anaerobic fungi. So we've gone to the Santa Barbara Zoo, which has sourced a lot of our samples from large herbivores that we have used roll tube based extractions to get single spores and then grow them up into cultures anaerobically, as you can see in the right there. And we've been able to generate single isolates from a number of strains listed there at the bottom I'll be talking about today. Hopefully what you can see is they're all a bit different, but they're all punching into that plant biomass with that rhizoid system and secreting powerful enzymes. And in fact, if you want to read more about the details of their enzyme systems, I'd encourage you to pull up a paper that we published in Science a few years back, which specifically shows that these fungi have the largest array of biomass degrading enzymes in the fungal kingdom to date. And so what we were trying to do is to leverage these new fungal isolates to find novel enzymes. One of the ways that we did that is to actually exploit regulation patterns so that we can use RNA sequencing, which I'll show you in the next slide, to actually figure out which gene sets are upregulated as the fungus transitions from glucose and more soluble sugars, where those biomass degrading enzymes are not needed, to things that are complex lignocellulose like reed canary grass. And in fact, in following the transcriptome remodeling during this process, so here's an experiment where we grew these fungi on lignocellulose and then peppered in some glucose and watched catabolite repression happen in real time, where we traced these blue transcripts which go down when sugar is added in a specific fungal isolate. And we see that the majority of the downregulated genes there were carbohydrate active enzymes, but there were a number of genes that we did not yet know what they did. And through guilt by association, we actually worked with some partners at the Joint Bioenergy Institute and Argonne National Lab to heterologously express and in some cases crystallize some of these candidate enzymes. So we've been able to identify novel biomass degrading enzymes like this glycosyl hydrolase 5 from Hyaromyces finis. And so just to give you a little bit of a taste of what else is in these genomes, so a big challenge in this field was not only isolating and just working with these fungi, but developing the technologies to really get a good look at their genome. And once PacBio sequencing came online, this became a lot more feasible. Here you see efforts from our group and also from the Mustafa El-Shahid's group for this Orponomyces isolate, that we now finally have some good draft genomes that are of higher quality than in the past. And they're pretty giant. So mining what's in there, one of the first things I'll point out is that we now have a recipe for codon optimization. So looking at open reading frames of these fungal genomes, they're quite AT skewed. So we've been able to use this in order to figure out a codon preference, which can help us then translate many of these enzymes from anaerobic fungi to other hosts and vice versa. One of the surprising things that we noticed is that the genomes of these fungi are home to a lot of novel membrane proteins that help them live their life in a competitive environment, specifically those that uptake sugars and other carbohydrates, but also drug extruders and so-called kind of guardian membrane proteins. And so these have been really helpful for us to actually put into monomicrobes like Saccharomyces cerevisiae. So here I'm showing you that we've been able to take a fluoride exporter, a putative fluoride exporter from the anaerobic fungi, and put that in the yeast Saccharomyces cerevisiae, and then recover fluoride export activity, and which is shown at the bottom there with a knockout strain that indeed does die when fluoride is around, but once we've introduced this anaerobic fungal transporter, it then recovers growth. One of the other things we noticed in the genomes is that these anaerobic fungi have a wealth of genes, biosynthetic genes for secondary metabolites. Here you see just 4 of those isolates and the numbers. This is just a pure bioinformatic prospecting, but it's rich in bacteriocins, which are actually peptides that kill bacteria, and type 1 polyketide synthases. And one of the reasons why we thought this made sense is because these fungi are there in such low numbers that they're probably distancing some of the bacteria and other widely abundant members of the rumen microbiome. So my student Candace thought of a really smart experiment where she, in fact, plated a lot of these anaerobic bacteria onto a plate and then added some fungal incubated medium, and saw that there was a characteristic clearing zone, meaning that we think we have some evidence that these fungi are producing antimicrobial compounds. And so just to wrap up, I hope I've shown you that anaerobic fungi are very cool. They produce a wealth of carbohydrate active enzymes, and they're certainly worthy of more investigation, and more groups should notice them. I actually didn't have a chance to mention this, but a lot of the Enzymes and other, even components of the membrane protein system have arisen through horizontal gene transfer with bacterial neighbors. Take-home message here is that in addition to the enzymes, there's novel transporters, gene clusters, and lots of even genetic elements that can be found in these fungal genomes that can be used to engineer yeast and really other organisms as well. And so with that, I will thank my group who's really done all of this work, and they are pictured here. You can see us on Twitter at O'Malley Lab, or visit us at our lab website, o'malleylab.com. And thanks to all of the funding and support that our lab has received over the years, which is indicated at the bottom. Thanks.
All right, Michelle, thank you very much for excellent talk. This is great. That's the question I have. So this is a kind of new area of exploration of fungi, right? Do we, with the 4 genomes, now 5 or maybe more, do we have enough references, or we need to sequence more?
I think we need to sequence more always. I think Igor would agree with me. I think it's the genomes that we've had and that others have generated with us as well have been really breakthroughs for understanding carbohydrate active enzymes, but there's still a lot from just the basic science standpoint of these anaerobic fungi that we don't understand, how their genomes are organized, how they're regulated. A lot of the information that we'll need to engineer them or edit their genomes in any way, we still need to sequence more genomes and figure out how to intervene in their function, and even just to model their normal physiology. So yes, we need more genomes.
Okay, great. And then with the more genomes, these are special kind of organisms, right? These are anaerobes, and I guess you're thinking about applied side, right? So how easy would it be to take enzymes from these organisms, from this species? One thing that I don't think you mentioned, they're assembled in cellulosomes, right? They're specially arranged to work in biorefineries, in sort of aerobic biorefineries. So do we need to think about special kind of setups?
Right. So we know from a sequence-based analysis that there's a bunch of carbohydrate active enzymes, including those that are associated together in fungal cellulosomes in these unique fungi. The ultimate applied idea is to take these enzymes and put them to work, right? Most of our biorefineries are not operating anaerobically, and so you might think that there's a bit of a discord there. What we've noticed is that when we've taken some of the sequences of these genes and put them into model workhorse platforms in industry like Saccharomyces cerevisiae, we can get many of these enzymes to function and to be catalytically active in the presence of oxygen. So it does seem like there's a route for translation that way. That said, every enzyme that we've tested, you know, some of them work and some of them don't. We don't quite understand why some do work and some don't. A lot of that we speculate is due to post-translational modification differences between the anaerobic fungi and other platforms, but that's, you know, purely a hypothesis at this point. So yeah, there will be some barriers to translating these to industry, but you know, yet another motivating reason why we need more research and more development on these fungi.
Okay, thank you. One last question is, are you able to preserve these strains for future studies?
Very important, especially because, as you saw in the talk, most of how we've gone out and obtained these isolates is in the environment, doing our own isolation. And, you know, it's important for a lot of reasons to try and preserve these so that we can establish a data bank for users, and so that we can, multiple people can do these investigations. So we and others have developed a cryopreservation strategy that seems to work well. It's basically a little bit of anaerobic technique combined with your conventional glycerol based freezing and on mature fungal sporangia, and that tends to work pretty well. We've been able to preserve strains for about 4 to 5 years and counting, although we're always nervous that we're going to lose viability of these strains. So, and if anyone's interested in obtaining these strains or learning how to extract your own anaerobic fungi, feel free to reach out to me by email. I'm happy to get back to you.
Okay, thank you, Michelle. Thanks a lot. So next, we'll hear about another large project, 1000 Fungal Genome Project, with a focus on plant pathogens in class Deuteriomycetes and whether we can use genomic information to predict lifestyles. So our next speaker is Dr. Sajid Haridas from the Joint Genome.
Hello, my name is Sajid Haridas, and I work as a data scientist at the Joint Genome Institute. Today, I'll be presenting a comparative genomics overview of 101 Deuteriomycete genomes. This large data set allows us to use machine learning techniques to predict lifestyles with high accuracy. The genomes presented here are available on microbiome, the fungal genomics resource hosted by the Joint Genome Institute, which currently has more than 1,700 genomes. Most of the genomes described here were sequenced as part of the 1000 Fungal Genomes Project, a large-scale community-involved project which aims to sequence at least 2 genomes from all known fungal families. This project was conceived several years ago, and we have sequenced over 600 genomes towards this goal. If you are working on an organism in an underrepresented family, we welcome you to submit your nomination using microbiome. As a national user facility, our services are available to the scientific community for free. So why Deuteriomycetes? The Deuteriomycetes is the largest class in the fungal kingdom and comprise an incredible diversity of lifestyles, many of which have evolved multiple times. Plant pathogens represent the major ecological niche here and are known to infect most major food crops and feedstocks. Previous studies have shown that the ancestral state of the Deuteriomycetes is probably saprobic, and there are many extant saprobes in the Deuteriomycetes. In addition to these 2 dominant niches, a couple of others are shown here. Deuteriomycetes consists of 2 major subclasses, the Pleosporomycetidae and the Deuteriomycetidae. In the expanded species tree for each subclass shown here, built using whole genome data, you can see that all nodes have greater than 99% bootstrap support. I have also mapped on the known lifestyles in the first 3 icons represented here. As you can see, the various lifestyles are not separated on different branches of the tree, but appear interspersed, suggesting several lifestyle switches. Previous studies have suggested that the ancestral state of the Deuteriomycetes was probably saprobic, and our analysis supports this hypothesis. So can we identify genomic signatures for these lifestyle switches? Looking at genome size and gene content in panels B and C here, we cannot see anything specific. On the tree here, I have indicated plant pathogens in blue and presumed saprobes in green as before. The two large genomes are the root-associated Zooxia rhizophylla, which is presumed to be saprobic, and Phencococcum geophyllum, which is ectomycorrhizal. In general, the saprobes show a slightly larger genome and more genes compared to the pathogens. So in order to identify particular genes correlated with lifestyles, we use machine learning approaches. Using support vector machines, which tries to separate categories across three-dimensional hyperplane with a gap that's as wide as possible, we trained on a set of features, including PFAMs, KZs, gene clusters, etc. And while functional annotation provided pretty good separation, gene clusters provided accuracies approaching 90%. The best results of greater than 95% accuracies were obtained using a combination of gene clusters as shown here. Using these gene clusters, we predicted the lifestyles of the Deuteriomycetes used in this study. The output of the program provides a probability of being a saprobe or a pathogen and hence the inverse diagonal. Here too, I have colored the pathogens in blue and presumed saprobes in green. The grays are other lifestyles. As you can see, the separation is near perfect. In some cases, such as STASP here, the apparent misplacement can be explained. This is one of Steganospora species. While other Steganospora species are known to be pathogens, this particular strain was isolated for its ability to oxidize minerals and therefore could be saprobic in nature. So what I have done today is introduce you to microcosm, shown you tools for comparative genomic analysis, introduce the 1000 Fungal Genomes project, where we have open call for nominations. I have described the Deuteriomycete genomes, which we have sequenced, assembled, and analyzed, and we developed machine learning tools to perform genome-based prediction of lifestyles. The work I presented here has been published earlier this year in Studies in Mycology. Please see this publication for additional details on methods and results. And I would like to thank all my co-authors for this manuscript. I will now take questions. Thank you.
Thank you, Sajid. Exciting story. While we're waiting for questions, I would like maybe to reiterate that this 1000 genome project is a great opportunity for anybody, any researcher in any parts of the world to nominate their species, right? To be sequenced and integrated in microcosm and be part of the studies like this. This is great opportunity. These genes that potential discriminators between lifestyles and predictors of the lifestyle. What do we know about function of those?
Well, all of them are currently without any functional annotation. Some of them show their presence across the larger tree outside the Deuteriomycetes. They are more or less restricted to the Pezizomycotina. We tried applying these machine learning techniques across the Pezizomycotina and we see the same trends. So there is something there, but that's the current state of knowledge, that we know pretty much nothing about them.
Okay, but then, so the concern, can the same features be used to beyond the Pseudomycetes?
Oh yes, so we see the same level of accuracy, at least in the Pezizomycotina where these genes exist. So we can use the same features to classify pathogens versus saprobes in the Pezizomycotina.
And another question is, can you further differentiate the pathogens in terms of biotrophs, necotrophs, etc.?
Not yet. So machine learning requires a large training set, and I think as we sequence more and we get a larger training set, that day will come. Hopefully with the 1000 Fungal Genomes project sooner than later. That's the hope.
Okay, and in linking to previous talk from Francis Martin, have you checked whether machine learning can distinguish symbionts?
That's something that we are trying out at the JGI, but we don't have an answer yet.
Okay. One other last question. I missed your comment on genome size. Did you say that saprobes have larger genomes?
In general, yes. Of course, some saprobe genomes are smaller than some pathogen genomes, but as a whole, saprobe genomes are larger and they have more genes compared to the pathogens.
Okay, great. Thank you very much, Sajid.
Thank you.
So we're moving to our next talk. Our next speaker, Professor Louise Glass from University of California, Berkeley, and she will talk about regulation of genes in a model fungus, Neurospora crassa.
Thank you very much. Today I'm going to be telling you about our systems of biology approaches to understand how filamentous fungi degrade plant cell wall material. And this is a collaborative project between my lab at UC Berkeley and also a laboratory, Philip Benz's laboratory at Munich, and also the JGI. Let's see. Okay, so many fungi degrade plant cell wall material, both ascomycetes and basidiomycetes, and we are particularly interested in this organism right here, Neurospora crassa, which degrades dead plant cell wall material. And that's because of the variety of genetic and biochemical tools available with this organism, the availability of a full genome deletion strain set, and the facile genetics. So the first thing we did was to determine how Neurospora senses and eats plant cell wall material. And so we exposed it to about 40 different polysaccharides. And so this is a heat map showing the transcriptional profiling results. And so you can see when you expose Neurospora to avocell, which is crystalline cellulose, it induces the expression of genes encoding enzymes associated with degradation of avocell. Similarly, pectin, you get a wide variety of enzymes, genes encoding enzymes that are induced, and that's because of the complex nature of pectin. Now, if you think about these complex polysaccharides, they can't be brought into the cell and degraded. And so that led to the following hypothesis that the organisms send out these enzymes as little scouts, and those degrade the plant cell wall material into disaccharides, monosaccharides, and oligosaccharides that are then transported into the cell and function as signaling molecules to induce these regulons. And so we tested the hypothesis by exposing Neurospora to a variety of small molecules, small oligosaccharides, mono and disaccharides, to see whether they could induce regulons associated with deconstruction of their parent polysaccharide. And so you can see, for example, with cellobiose, it induces not only the enzymes and genes involved in cellobiose utilization, but also cellulases and LPMOs. So this is what the organism senses intracellularly to turn on this regulon. And so we were very interested in the regulatory network associated with inducing all of these different regulons to these complex polysaccharides. And so we took the following approach. We first identified transcription factors in the genomes. There's around 350 in Neurospora. We filtered those transcription factor candidates based on our profiling data on a variety of those carbon sources and also nitrogen, phosphorus, and sulfur sources. We did expression profiling of transcription factor deletion mutants under these different nutrient conditions. And so basically we just went to our -80, pulled out the transcription factor deletion mutant because we have a full genome deletion set, and then did the hierarchical clustering. And then finally, we used a technique called DAP-seq to identify the direct targets of these transcription factors. And this is an alternative to chromatin immunoprecipitation. And so DAP-seq was developed by Ronan O'Malley when he was a postdoc. He is now at the JGI, and this is a function provided by the JGI. And so what is done is the genomic DNA of your target organism is sheared, and adapters are ligated for PCR. Your transcription factor is made in vitro and attached to an affinity tag, which is then attached to a column, and your adapted DNA is run over that column, and anything that binds to that column is PCR amplified and sequenced. And so using this method, we were able to identify new transcription factors and their target genes based on a combination of expression profiling of the transcription factor deletion mutants and DAP-seq. And so this is just a circus plot showing the direct targets of all these transcription factors associated with carbon utilization. Now, if you think about fungi in nature, they live in a carbon-rich environment, but a very nitrogen-poor environment. And so they have to be able to integrate signals both from carbon and nitrogen to optimize their internal metabolism for optimal growth. And so on the top here, you can see the genes that have been identified genetically that are associated with nitrogen utilization in filamentous fungi. And so what we did is we took wild-type Neurospora, used the same approach as with our carbon utilization. We exposed Neurospora to a variety of different nitrogen sources, went into our transcription factor deletion set, and evaluated their direct target genes by DAP-seq. And from this, we were able to identify direct target genes of known transcription factors and also identify unknown transcription factors. And this particular transcription factor, AMN1, integrates mannose utilization and also catabolism of amino acids, indicating, again, this crosstalk between regulatory pathways. And an important component of both of these studies is that transporter genes are often a direct target of these transcriptional regulators. And that makes sense because those small molecules that are transported into the cell are the signaling molecules that the organism senses. And so these are the people that did the work. I just want to highlight Vince Wu and Lori Huberman and David Kabel, who did the ENCODE project along with our collaborators Igor and Ronan at JGI for both the carbon and nitrogen utilization. And so thank you very much.
Thank you, Liz. Excellent talk. Another example of why we shouldn't stop at sequencing genomes, but go to the next step, right? Genomes are not enough to understand biology. While we're waiting for questions, maybe I'll start with my question. So, to which extent do you think the transcription factors from Neurospora crassa and regulons they work on conserved across other fungi? How this information we're getting from your work is possible to spread over the tree of life?
Well, an example that I can give, there are two really. One is the CLR1, CLR2 transcription factors that we identified in Neurospora that are essential for cellulose degradation. They are also important for cellulose degradation in Aspergillus nigilans, which is a member of the Rochiales and probably the most distant group from the group that Neurospora is in the Ascomycete species. In addition, the nitrogen regulators that we characterize both transcriptionally and by DAP-seq also have conserved regulons and functions in Aspergillus nigilans, where they've been well characterized, and also in Neurospora crassa. Those are NIT4 and NIT2. So I think that for nutritional signaling, there's probably nuances based on the lifestyle of the organism. But for many of these major nutritional signaling components or transcription factors, they probably are conserved. And so, for example, this new transcription factor that we identified that's involved in nitrogen regulation also actually has a role in aromatic amino acid regulation in Saccharomyces, so even more distantly related. Basidiomycetes, I think, are a different question, and I'm not sure about the conservation of Ascomycete regulatory networks into Basidiomycetes.
In which case, how it is to just go to another target and repeat the experiments that you've done on Basidiomycetes or something else, very remote from the Neurospora crassa?
Yeah, so I think the advantages of doing some of these preliminary experiments in Neurospora is that then you can start to look at evolution of these regulatory networks by starting to look at organisms that are closely related to Neurospora and more distantly, and see how they evolve over time. And so I think using a combination of DAP-seq and RNA-seq, and again, for us, transcriptional profiling of the deletion mutants was really informative in terms of getting rid of false positives and false negatives, that we can start to understand how these transcriptional networks evolve over time and in organisms that have different lifestyles, dung fungi versus leaf litter fungi, pathogenic fungi versus saprobes fungi. And I think that'll be a really interesting study to extend that.
And deletion mutants to this extent available only for Neurospora crassa or for some other, that would be the bottleneck, I assume, right?
Yes, but I think with the CRISPR technology that actually making some of these mutants, and maybe not clean deletion mutants, but I think making some of these mutants is now more technically feasible than it has been in the past. And it could be that only, you know, for Neurospora, we have all the transcription factor deletion mutants that are viable, but it may be that there are select and critical transcription factors that could be targeted in species rather than, you know, sort of a survey approach.
Okay, great. Thanks a lot. So the comments about great talk, and yeah, thank you for sharing this with us. So next speaker, last but not least, Dr. Scott Baker. He's the lead of functional and system biology, part of Environmental Molecular Sciences Lab and other user facilities similar to JGI at Pacific Northwest Laboratory. And he will describe another large-scale sequencing project focusing on a single genus.
Hi, I'm Scott Baker, and I'm excited to be giving this presentation on the whole Aspergillus genus genome sequencing project, and I want to thank the organizers for the opportunity to give this talk. So I first want to start with an acknowledgement. What I'm going to show you over the next few minutes would not have been possible without a team of teams. My co-PI for this project was Mikael Andersson. He and his team at DTU led the analysis that I will talk about. And the Fungal Genomics program at JGI, led by Igor Grigoriev, was responsible for the sequencing and gene modeling and some analysis for all the genome sequences. Ronald DeVries and his team at the Westerdijk Institute generated the growth profiling data. We aim to sequence approximately 280 genomes, one type strain for each species in the Aspergillus whole genus genome sequencing project. We have been analyzing genomes by sections within the genus, the first being Aspergillus section Nigri, which includes biotechnology relevant species, including Aspergillus niger. Here you can see a multi-gene phylogeny of the section, as well as various genome statistics such as gene model counts for each species. And here's the gene model counts. You can note that they vary across the species, even though they are closely related. Prior to this project, we noted that the genome of Aspergillus niger encoded a gene cluster for the biosynthesis of the mycotoxin fumonisin, which at the time was only known to be produced by species of Fusarium. Notably, a fumonisin biosynthetic cluster is also found in Cochleobolus heterostrophes, a Dothrodiomycete. And what I'm illustrating here is really the utility of genome sequencing across a diversity of fungi. When we do that, we see that there are some shared characteristics even across phylogenetically different fungi, or distant fungi. As we sequenced multiple numbers of Aspergillus section Nigri, we were able to assess the presence and absence of the fumonisin biosynthetic cluster. Here we show the full cluster is found in multiple species. However, truncated clusters are also found in other species. And a second polyketide synthase of unknown function is found adjacent to the primary fumonisin PKS. So there is still much to learn about fumonisin and the biosynthetic clusters that encode the enzymes responsible for making fumonisin. Here's a bird's eye view of secondary metabolism in Aspergillus section Nigri. Of note are the large number of potential backbone encoding genes compared to relatively small number of secondary metabolites that have been identified in these species. And those are indicated on the right of the phylogenetic tree. Moving on to section Flavi, we see another phylogeny based on multiple predicted proteins. This part of the Aspergillus genus contains important plant pathogens as well as koji fungi important in food production, notably Aspergillus oryzae and Aspergillus sojae. In this phylogeny, the placement of Aspergillus sojae is not thought to be correct, hence the arrow, the red arrow. And that's based on differences in gene calling algorithms used in our study as compared to previously published Flavi genomes. Similar to section Nigri, we see a high-level view of secondary metabolism in section Flavi. Again, there's tremendous potential chemical diversity indicated by the number of predicted backbone genes. On the left is a heat map of growth profile data for each species within Flavi on certain carbon sources, and on the right is a comparison of Enzyme sets related to plant biomass degradation encoded within each genome. The take-home message here is that the encoded Enzyme diversity is only part of the equation as to growth performance. Regulation of these enzymes is really a critical determinant. I'll end with a snapshot of secondary metabolite diversity in Aspergillus saccharnusti. The take-home message is that the more closely related species, the more similar their secondary metabolite encoding gene repertoire. However, even among closely related species, there's considerable diversity. So I'll summarize by saying that this genus genome sequencing project gives us a snapshot of genetic and chemical diversity within Aspergilli at single species resolution. That also allows future studies to delve in and look at really the details. There's a tremendous diversity of secondary metabolite biosynthetic encoding genes, and regulation of all the nutritional digestive enzymes is a critical component of how different species grow in a variety of carbon sources. Next up are whole genus projects for trichoderma and penicillium that are currently underway at the JGI. Thank you.
Thank you very much, Scott. Excellent talk. So we're getting close to 300 genomes sequenced.
Yes.
Do you feel, from the past, we heard that the idea of Earth's Biogenome Project is to sequence every species of eukaryote in a few years, and we're currently at 3% or something around that number. Do you think it's a feasible strategy for going to other genera? What would be the bottlenecks? What do you see as the primary issues? Does it make sense to do this for other groups?
Yeah, actually, I do think it makes sense. This has been a really exciting project because you get to this single species resolution. I think that I don't know if it's not really a bottleneck so much. One of the bottlenecks is obviously prepping all the materials. It's a lot of material and strains to grow and prep and make sure that you've got the right species growing and the sequencing. But I think if I had it to do all over again, and there will likely be some sequel projects to this, I'd like to do more sequencing projects where not only are we doing the whole genus, but we're zeroing in on some species and doing multiple strains just to look at the diversity within a single species. I think that is the next step for this kind of project. And it would be very exciting. You know, what limited data we have, I think there's some interesting results from that.
Okay, great. And so secondary metabolism, secondary metabolites, available for Aspergillus strain was one of the sort of key feature of this project that allows to annotate some of the groups of genomes. Are we in a similar situation with penicillium or trichoderma, the next targets that you mentioned?
I think Aspergillus, penicillium, there's been a number of well-characterized metabolites from that genus. So I think it will be more similar to Aspergillus. I haven't done a deep dive into all the different metabolites that have been characterized yet. I think there's less known about the specific secondary metabolites and their associated gene clusters in trichoderma. Certainly there are some that have been identified, but I think you hit on another aspect of this. We can't just keep sequencing genomes. We have to, do other kinds of assays. And, in particular for fungi, I think the secondary metabolism is really exciting and important because there's such a chemical breadth of compounds that these organisms produce.
Okay, and so the last question, are there a correlation between gene content and either niche or biogeography? And let me add to that. And do you have any concerns about contamination during sampling?
Okay, so the first part, the gene content, we do see variability across these genomes, even closely related ones. I don't know that we've really looked at that in depth to be able to give an answer. As far as cross-contamination, I think that it happens. It's the exception though, rather than the rule. Certainly once you've sequenced something, you have a better idea of what it is. And for the, 99% of the isolates we've done, we've been careful enough, and it was really Mikhail's team at DTU was very careful and kept everything straight, and it worked out quite well.
Okay, great. Thank you, Scott. I would like, so we're running out of time. I would like to thank all the speakers of this session for excellent presentations. And I would like to thank also all the participants who join us for this session. And so we can continue discussions during a poster session. So there is a link shared in questions. Please join us there for more details about these projects and others. And there are 7 posters in the fungal sessions to discuss too. All right, thanks a lot.